In the previous video, we discussed replication, a process in which two molecules of DNA are made from one molecule of DNA in the nucleus. In that process, we discussed several enzymes that are in charge of this replication process. We discussed that helicases are in charge of opening up the hydrogen bonds between the nitrogen bases. We said that G and C are going to separate, and AT. We also discussed how we use primases, that is to bring small little primers for the synthesis to work. And that reaction is happening simultaneously in the leading strand. So we see here one primer for the leading strand. And we also have that reaction happening in the lagging strand, making small Okazaki fragments. We also say that DNA polymerase is in charge of making phosphodiester bonds with the incoming activated nucleotide, and is happening simultaneously in the two strands. So we see the two parts of the enzyme that is working both at the same time, one making a continuous strand, the second one making a discontinuous one. Now we discuss transcription, the process in which we are going to make a molecule of RNA from the template of DNA. So we have a molecule of DNA that is all going to open up. One will be carrying the information, and one of the strands only, that is the template, will be making a sequence that is nearly identical to the sequence of the coding strand. We have seen the molecule of RNA being used during replication when we have primers. But why is that we're now making changes? We will have two major changes. The first thing is we are no longer using 2' deoxyribose like in RNA, like in DNA. Now we are using regular ribose, and now we are replacing. So we say in DNA we have ATGC, now we have AUGC. So we are replacing one of the nitrogen bases, and the sugar here was 2' deoxyribose. Now we are using just regular ribose. And why is that? Because we want to keep the information when we have the 2' deoxyribose by the use of an enzyme called ribonuclease A. That enzyme can do the cleavage of phosphodiester bond, and that's what I'm showing you here. Why is that in DNA we use the 2' deoxyribose? Now we are using just regular ribose. And that is of major importance. During transcription, we are making a molecule of RNA that is made in the nucleus and is going to exit carrying the information from the nucleus to the cytoplasm, where it's going to join transfer RNA, ribosomal RNA, and messenger RNA to do what is called gene expression, that is to make a polypeptide, to make peptide bonds. So we are changing from the language of nucleic acids, phosphodiester bond in replication. We do transcription, keeping the language of phosphodiester bond in the nucleic acids of RNA. And then in translation, we change the language of phosphodiester bond to the language of peptide bonds between amino acids, or the language of amide bonds. But the molecule of DNA is a really long molecule. So how does RNA polymerase know when to start the synthesis of an RNA molecule to make a protein? And the answer to that is what is called promoter sites. In the templates of DNA, there will be some sections that are found to be consistent, where the opening of the molecular DNA will happen by the use of helicases that are opening up, so that we can have a synthesis of a RNA molecule. As soon as the synthesis of RNA begins, the molecule of RNA is going to be capped. What is the meaning of the word cap? A cap is a modification of the nitrogen bases by inclusion of methylation. So we say that you have a nascent RNA molecule, so we call that the nascent RNA molecule that is going to have modification in some nitrogen bases. And when the synthesis is complete, to release that RNA molecule that will go to the cytoplasm, an addition of adenine will also happen to that RNA molecule. So what is the capping of a molecule of RNA? The first thing is that we will have methylation of the sugar unit and also methylation of the nitrogen bases. A ribonucleic acid is called a primary transcript or premature RNA. This ptRNA is modified by post-transcriptional modification to produce messenger, ribosomal, and transfer RNA. Some of this processing takes place in the nucleus and some takes place in the cytosol. The purpose is to stabilize and optimize a specific RNA for its function during protein synthesis. In prokaryotes, genes are found in the molecule of DNA to be continuous to produce different proteins from each gene. In eukaryotes, we will have that only 3% of the sequences of DNA will codify for a protein. So we will have what is called intervening sequences, and in those intervening sequences are called introns that are not going to codify for an RNA molecule. We will say that introns are going to be removed. That's what it's called, and then that's what is called splicing. So we are keeping the exons. Exons are going to be expressed. So we have from a long DNA molecule, some introns are removed, the gray part, and then the exons will be joined together by splicing. And after splicing, we get a mature RNA that can codify for the synthesis of protein. After RNA is made during transcription, it's going to leave the nucleus, and then we're going to have three different kinds of RNAs. We will have the transfer RNA, that this is the one that carries an amino acid. We need amino acids to be put together and make polypeptides. We have the messenger RNA that carries the sequence of what will be the sequence of amino acids that needs to be put together. And then we have ribosomal RNA that when it combines with lots of proteins is going to make the ribosome, where two tRNA are going to join two amino acids to make a peptide bond.